Okay, now we come on to the main process of transcription. See here, transcription, you can see a transcription bubble being formed over here. Here the transcription bubble is formed where the sense strand and the antisense strands are separated and give a bubble kind of appearance. Now this is the RNA polymerase which is already present and unwinding has been done. Clear? Now what happens? The 3' to 5' strand is being used as a template strand and the newly synthesized messenger RNA is being produced. Okay, so this is how this is produced. This is produced by complementary base pairing, and then the polymerase, RNA polymerase keeps on moving, just like movement over the chain. The runner moves on the chain, the RNA polymerase runs on this chain of DNA, and the the transcribed region again winds up, and again the region to be transcribed keeps on unwinding to provide a region to be transcribed. That means forming a transcription bubble. So this is the major process. We will discuss it stepwise in 3 steps. The first step will be initiation. See what happens in the process of transcription. The single stranded RNA molecule is referred to as messenger RNA. You all know this. So the process of formation of RNA from DNA is called as transcription, and this involves the formation of RNA strand on the DNA template. I think this much is clear to everyone till now. When initiation is required, that means when the process of transcription is being initiated, the sigma factor of the RNA polymerase binds to the promoter region. See the diagram. The promoter region is the recognition sites on the DNA. This is the DNA double helix. This is the promoter region. Now, the sigma factor of the RNA polymerase binds to the promoter region of the gene and produces the uncoiled structure, or what we call as the transcription bubble. So, when the RNA polymerase binds to the promoter region, it tells the DNA to separate out. That means to form a bubble kind of structure which we call as transcription bubble. The RNA polymerase helps in the formation of first phosphodiester bond. So the process of initiation has 2 steps. First is the recognition of the promoter region and binding to the region which has to be transcribed, and then forming the first phosphodiester bond. The antisense strands makes available the RNTPs, the NTPs which are required, ribonucleotide triphosphates such as ribonucleotide ATP, ribo-R, RUTP, RCTP and RGTP. Here instead of thymine, uracil will be required, because RNA is being synthesized. Just compare it with the process of DNA replication and it will be clear to all of you. Now what happens once the transcription bubble is formed and the first phosphodiester bond is formed, that is 2 phosphodiester bond will be formed between 2 nucleotides. That means one nucleotide has come, another nucleotide has come, and the first phosphodiester bond has formed. Here the process of initiation has completed. Okay, so initiation is complete. Now the next process followed by this will be elongation. Once the first phosphodiester bond has formed, now the chain will elongate. See here the diagram. Elongation, you can see elongated RNA, messenger RNA, a long messenger RNA has formed, because now as one nucleotide has been added, certainly again second, third, fourth, fifth, sixth, and the chain continues and the messenger RNA is formed. So the elongation phase begins when RNA polymerase leaves the promoter and continues to transcribe the template strand. Till now, in the initiation process, RNA polymerase was just holding the promoter region and giving the signal that from here the gene has to be transcribed. But as soon as the transcription has started, RNA polymerase leaves the promoter region and moves through the bubble, and continues with the process of transcription by elongation. Clear? Just understand here the coding region. I have made this here coding region. If the coding strand has A, T, G, A, and so the template strand will be having T, which is complementary to A will be complementary to T. Okay, these blue strands are DNA and the pink strand is the newly synthesized messenger RNA. So you can see the newly synthesized messenger RNA, which is pink in color, AUG, is actually ATG. I told you, instead of thymine, what will be present in RNA? Uracil. So this is the complementary base pairing of the template strand, and template strand is actually complementary base paired with the messenger RNA, but messenger RNA is the exact copy of the coding strand or the sense strand. That is what I was telling you in the previous diagram of sense and antisense strand. Now in the process of chain elongation, the core enzyme keeps on adding the correct ribonucleotide triphosphates and the chain elongates in 5' to 3' direction. You can see the newly synthesized messenger RNA is 5' to 3'. So the chain keeps on elongating. The rate at which RNA polymerase function is 40 nucleotides per second. So it's a very fast rate, and that is at 37 degrees centigrade. Okay, so that and what happens after that? The transcribed region of the antisense strand reassociate with the sense strand to form the double helix again, so that it unzips and again it zips and binds with each other. Okay, clear? See, one thing I want to make very clear here, that whatever we are discussing in transcription and protein synthesis, we are discussing about the prokaryotic cell. Only you have to study about the prokaryotic cell. Whenever there will be a change, I will let you know. Okay, so here the elongation continues. Now after the elongation, after the required amount of DNA has been transcribed, the next step comes is the stopping, that is termination. But before the termination, I want to show you one diagram where you can find out two transcription bubbles at a particular time. See here, the two transcription bubbles are formed here. This can also be the condition. Just to make you aware, I have put this diagram here, because here you can see one transcription bubble is formed here, 5' to 3' direction, the newly synthesized, it is moving in this direction, opposite, and here this transcription bubble is moving in this direction, opposite. So when two genes which are separated by a certain non-coding region, certain region which is not to be transcribed, then what happens? Two RNA polymerase may bind at different places and continue with the process of transcription simultaneously in opposite direction or in same direction as and when required. Clear? So this diagram is clear to everyone. That is, two transcription bubble or more than two transcription bubbles can also be present.